Hello students, how are you? As always, generally we start the class on a very positive note. So again, we are doing the same thing. And as you can see on your screen, the key to success is to focus on goals and not obstacles. These are very hard times for everyone. Me as a teacher, you as a student, everyone. It's a very hard time because of this COVID. But you don't have to focus. Why? Because that's not your goal. And what is your goal? Your goal is to become a doctor. And so these are just few obstacles. Don't focus on them. So let's focus on our goals and let's start the today's topic, that is digestion of starch. So the today's topic is a small topic. It is digestion of starch. Okay, so what are we going to do in this first? The first thing that we're going to do is first we'll review the structure of starch. Then only you will be able to understand how the breaking down of this starch is taking place. Okay, so we'll go very slowly. Digestion of starch in one, digestion of protein in one video, digestion of fatty acids in one video, then absorption, how are these getting absorbed, and then the neural and chemical regulation also. So if you want to study or stuck in somewhere in digestion of starch, you don't have to see the whole 2 hours video, just a small 15-20 minutes, half an hour video. You see it and then you solve the question wherever you are stuck. Okay, so let's start with the review of starch and then we'll do the digestion. So these are the two things that we are going to do. So suppose your mama has made a very yummy sandwich today. So what is your mama giving you when she's giving you sandwich? You know that you're eating starch, right? So starch is formed by two forms of glucose polymers. What am I saying when I'm saying that the starch is formed of two forms of glucose polymers? One is the linear one and the other one is the branched polymer. So now what is this linear polymer? If you want to understand with the structure, linear means that these glucose molecules are arranged in a linear manner, right? And these glucose molecules are arranged in a branched manner when we are talking about the, see, one branch here. So what are these blocks that I am making? These are the, what are these? These are the glucose molecules. Okay, so this linear is what? This is known as amylose, and this branched polymer is amylopectin. Okay, and these bonds are alpha 1,4 glycosidic bonds. Similarly here also alpha 1 to 4 glycosidic bonds, right? And this branched, where it is branching, it is alpha 1 to 6 glycosidic bonds. So I just wanted to tell you this structure so that when you're doing the digestion, it's easier for you. And what is this? One thing that I am drawn, this is glucose molecule. Okay, understood? Now let us see the question. Let us try to understand this question. What happens when starch is ingested? When we eat starch, what happens? It goes in our mouth, right? And there, two types of breakdown will take place. One physical breakdown and one chemical breakdown. So the physical breakdown will happen by what? Very easy. Jaws, teeth, right? We have done all this structure. Tongue, and the chemical will be by the salivary glands or salivary amylase. And what will this salivary amylase do basically? This salivary amylase will hydrolyze or simply break down which bonds? Alpha 1,4 glycosidic bonds. Right, that's why I had told you that. So what I'm trying to say is that if this is a glucose polymer with alpha glycosidic bonds, what will happen in the mouth? It will become short chain. Basically, what is happening? Basically, this breakdown is taking place. Okay, so it is, what is this happening? This is basically breakdown happening. Easy to understand? Now, let us see what is happening more. Then what happens? Then it goes to stomach. And what happens in the stomach? What is coming in the stomach? What is coming in the stomach? Partially digested. Partially digested starch is coming in the stomach. So basically, what is partially digested starch that I'm talking about? I'm talking about oligosaccharide and short chain polysaccharide, right? So the polysaccharides have become short chain or oligosaccharides. Now what will happen when it comes to the stomach? Now this poor amylase, what happens to this? It is inactivated. What is happening? It is getting inactivated. So basically, what are we trying to say? We're trying to say starch digestion does not occur in stomach. Okay, then nothing happens for uselessly in our body. What is the stomach doing? The stomach, what is the stomach doing? Will only mix the contents. It will only mix the contents and send it to small intestine. Okay, it is sent this to, and now the small intestine, here the maximum digestion will take place. Maximum digestion of starch will take place. Okay, so now we know that in the intestine, partially hydrolyzed starch is coming. So let us draw these enterocytes. What are enterocytes? Enterocytes are the absorptive cells found in small intestine. We have done this when we are doing the structure of small intestine. Okay, so these are what I have drawn. These are enterocytes or absorptive cells. Okay, absorptive. This is the lumen and here is the bloodstream. What is this? This is the blood stream. Okay, right. So what will happen here? Let us see. Okay, what will happen here in the enterocytes? So when the food is reaching intestine, the food is in which form? The food is, not the food. We should not say food. We should say starch. The starch is in which form? It is in the partially digested form. Okay, please remember this. When it is reaching intestine, it is in the partially digested form. So when we'll come to this diagram again, let me just tell you one more thing. When starch, we are talking about starch, but we can say food here. When the food reaches intestine, pancreas भी तो है, pancreas will release, we have done pancreas. Pancreas will release what? They will release alpha amylase, like the salivary glands. And what will happen? The hydrolysis will take place, or the breakdown will take place of what? Alpha 1,2,4 glycosidic bonds. Okay, so they are getting further digested, further breakdown. Okay, so now what is happening in these enterocytes? Got bored with the black. Let us take purple. Now what will happen in these enterocytes? Enterocytes have brush border enzymes. Okay, these have brush border enzymes. So like we have maltase. We have here maltase. Okay, now let me change the color. We have sucrase. These are what? These are enzymes that I am showing. Sucrase or isomaltase. Okay. So this purple thing that I have drawn is what? This is maltase. And this green thing that I have drawn is what? This is sucrase or isomaltase. These are what? These are enzymes. What will these enzymes do, right? The question is, what will these do? So let us see what are these do. These things are doing. So this maltase will be converted to maltose. And what is maltose? It is 2 glucose molecules. Okay. And if we talk about sucrase or isomaltase, what will it do? It will, now please see, it will work. Hey, where are you going? So what will it do? It will work on both. It will work on alpha 1,4 glycosidic bonds. So when it works on alpha 1,4 glycosidic bonds, what happens? The linear bonds are broken. So when it works on this, what happens? Linear bonds are broken. Okay. Remember when we did the structure of starch? And it will also work on alpha 1 to 6 glycosidic bonds. And now what is happening? The branched bonds are broken. Okay. Right? Understood? Very easy to understand. Now let us see what is happening here. Right? In these enterocytes, what is happening? So this is what there, this partially digested starch is there. So we have this kind of structures, some, you know, like this, some are more linear, more big. This is what? This is the lumen where, and these are the cells. So still some are big, some are branched because of these alpha 1 to 6 glycosidic bonds on which this sucrase will act. So there is this partially digested food is there. Okay. So now what will happen? These all will be broken down into what? They all will be broken down into small glucose molecules. So what is happening? These all are getting broken down into small glucose molecules. So what are these red things that I am drawing? These are glucose molecules when after the breakdown. Okay, they are not present in the lumen, they are after the breakdown. So this all is getting broken down into this glucose molecule. Now we also know that the lumen also has sodium ions. I'll tell you why I'm telling you about this sodium ions, right? We also have this sodium ions, okay? Now there is a very interesting thing present on this, where should I draw? On this apical surface. And now you know by the structure what I'm drawing. These are the sodium glucose transporter. These are the sodium glucose transporter. So for every glucose molecule, two sodiums will also enter. So now what is here inside? Two sodiums have entered for one glucose molecule. Now what will happen to this glucose molecule that is in the enterocytes? These are what? These are enterocytes or absorptive cells. Enterocytes or absorptive cells. So now what will happen to these glucose molecules that have come here? Something needs to be done. They can't just roam around, and glucose is what? It is energy. And there is a bloodstream. So what will happen? These glucose molecule will go into the bloodstream by again a transporter. So there are names for these transporter which will be too much for you. So you just remember that there is this, I should make it with a different color. So this is what? These are transporters, and by the help of this the glucose molecule is going in the blood stream. And what will happen to the glucose molecule? What will happen to the glucose molecule? Tell me, think. The glucose molecule will be used as energy by tissues or stored as glycogen in liver. So understood, right? This is how it is getting digested. Now let us see. Now let us see what happens to the few stubborn and resistant starch molecules which say we will not get digest. So these, what happens to these resistant starch molecules? They reach to the colon or the large intestine, you can say. And what is present in large intestine? There are, what is present? There are gut bacteria, right? There are what? Gut bacteria. Can you name few gut bacteria? We are doing this all gut bacteria when we're small even. So E. coli, or one more, Streptococcus bacillus. What will this gut bacteria do? What will this gut bacteria do? This will change it into short chain fatty acids, right? Like this, and it will be subsequently used subsequently used by the body, right? So we have done the digestion of starch. Let us see what happens. We did started with digestion of starch. We did the structure of starch, and then we did the digestion. So we studied that there are two forms of glucose polymers. One is linear, the other one is branched. This is called amylose, and this is called amylopectin. These bonds are called alpha-1,4 glycosidic bonds. And here also in branched, these are alpha-1,4 glycosidic bonds. But these branched bonds are alpha-1,2,6 glycosidic bonds. Okay, so then we tried to answer what happens when the starch is ingested. We tried to answer this. So in the mouth, two types of breakdown will take place, physical and chemical. And in the chemical, the salivary amylase will hydrolyze or break down these alpha or linear bonds, and small chain glucose polymers are formed, and then it goes to stomach where no starch digestion is taking place. Now what happens to the amylase? It is getting inactivated. So now the stomach will only mix the contents, and then we did about this. This is basically what these are the absorptive cells with brush border enzymes with, and then we saw that how these partially digested starch is getting broken down into glucose, and glucose is entering enterocytes, and how it is going into the blood stream, and finally it is being used by the body or being stored as glycogen. And resistant starch molecules, we said, by the gut bacteria, they have been broken down and subsequently being used by the body. Now for your exam point of view, I'll give you one chart that will actually help you to remember few things. So the heading says enzymes, from where those enzymes are coming, substrate on which those enzymes are adding, and what are the end products. Suppose, let us start with salivary amylase. What is the source? Salivary glands. What is the substrate? Starch or polysaccharide. What are the products? Maltose, maltotriose, alpha dextrins, and maltase. Now let us go to the other enzymes. Maltase, from where it is source is small intestine, you see, no stomach, okay? And the substrate for maltase is maltose, and what is the end product is glucose. Now sucrase, when you see this chart, you will just get a summary. Sucrase will act on sucrose, and it will be broken down into glucose and fructose, and then we can do lactase. The source is the small intestine. It acts on lactose and glucose plus. You know why I'm giving you this? Because when we'll do absorption, then you should know all these end products. How are these getting absorbed? So enzyme, source, substrate, and products. Enzymes, source, substrate, and product. So students, we have done digestion of starch. So subsequently we'll be doing digestion of proteins and other things also. So please study hard, and I'll give you questions also. So probably next is digestion of proteins. Hope you understood digestion of starch nicely. So please stay safe, stay at home, study hard, utilize your time. Bye guys, have fun.